Okay, so far we are focusing on concept theories, genetic mechanisms related to a single organism. So we have learned taking one plant, its phenotype, and taking another plant, its phenotype, and mixing them in a breeding experiment. What would be the outcome of the offspring like this? Now, in this lesson, we are going to learn genetics in a population. Now, when we call population, the definition of population is a number of individual in a species at particular time in a particular place. So, one species, how many numbers in that species? What is the time? Where the place? Example, human population in Sri Lanka in 2023. So when we say human population, human is a species. In Sri Lanka is the geographical location. In 2023 is the time. So that is the right way of defining the population. Again, we can also define population, not express the population, the human population in Colombo district in 2023. So then we only restricted to Colombo. So our genetics concern about this kind of a population. It could be in a city, it could be in a country. We can equally apply this kind of concept to understand some kind of to get some kind of information. For example, when we are learning cystic fibrosis, I showed you cystic fibrosis is common in certain countries like UK. So it's a UK population we take as a whole, and then we're going to study the cystic fibrosis. In that case, practically, this is useful to evaluate certain percentage, certain number in terms of genetics. What is the uh, size of the population who has this kind of a genetic background? But we have a problem. The problem is like this. Uh, Before we come to this Hardy-Weinburg concept, so we're going to learn actually here only one concept, Hardy-Weinburg principle, Hardy-Weinberg equilibrium. We don't want to learn. We are not going to learn many other concept in population genetics, which are not in the syllabus. Okay. Now, when we consider the evolution in Unit 3, we have learned evolution is change of the phenotype of a species over a long period of time from generation to generation. Change of the phenotype. That means phenotype is changing, genotype is also changing. And these changes are inevitable. We can't stop these changes. All the organisms, they are subjected to evolution. So phenotype is changing, genotype is changing. Now, when we consider this concept, all organisms are evolving, they are changing according to the environmental conditions, and their phenotypes are changing, their genotype is changing. Then we take a conclusion, genotypes are not constant. So when we consider genotypes are not constant, we can understand it like this. We take the flower color which we learned before. So we take a population. They're having flower color, purple and white. Then we can understand the amount of purple and white. There is certain amount of ratios. That means if it is according to Mendel genetic, dominant and recessive is there, then the dominant one is more, recessive one is less. Why is that? According to the probabilities, if we get the equal chance to everyone, so having a phenotype like capital A, simple A, they are both express a purple color. Let's say blue color, because here dominant alleles and here one dominant allele that can make a blue color. And here again a recessive one. The recessive one give the white color. So if I want to show this one in a diagram, so I can show, okay, this is the purple and this is the white flower. Right. Now let's leave this population, and it goes maybe many years, like it takes time pass, right? So if the time pass, we can apply the theory, yes, population is evolving, they are changing. We can't keep them like this always with the time. Then what if we see this population after many years later like this? These are the white flowers and this is the blue flower. Now phenotype shifted to a blue color than the white color. It was here dominating blue, now here dominating white. Dominating mean number, right? More white flowers with the time and more blue flowers in the past. So we can give any justification. Probably blue flowers are having some disadvantage when the environmental change. Then the white flower get this advantage being color white and they increase. This is very simple example to tell uh, that the phenotypes are changing. But these are example, not true thing. I'm not going to argue differently. Just only learning purpose, we're going to take this example. What we wanted to learn is this. Let's assume the genotype of these flowers. This one is a capital A, capital A, capital A, capital A, simple A, capital A, simple A, capital A, capital A, capital A, capital A, capital A, simple A. This one is simple A, simple A, simple A, simple A. Now, I wanted to find the frequency of capital A, frequency of simple A. The frequency is how many time existing. So I count total, total alleles, right? So total alleles. Total alleles 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. 8. So The 8 phenotype means 16 total alleles. Total alleles 16. Then how many alleles for blue color? How many capital A's? Capital A, 1, 2, 3, 4, 5, 6, 7, 8, 9. It's 9. Then if I want to do a simple simple A's, then it should be 16. 1, 2, 3, 4, 5, 6. Now, if we write like this, total alleles, and then over it, how many capital A or simple A, and we can further simplify this, 9 divided by 16 and 6 divided by 16, right? So it should be 9 divided by 16, 0.56, and 6 divided by 16, 0.3. Okay.
So even the purple flower can also have simple leaves.
Yes, these two, right?
Yes.
Yeah. So this one is a white. But what I take, I count separately how many capitals A, how many simple A. So this number is not correct because I had to recalculate the capital A's 1, 2, 3, 4, 5, 6, 7, 8, 9. And simple A 1, 2, 3, 4, 5, 7, right? So it should be 7. I miss 1. Should be 7. So 16, 7 divided by 16. 7 divided by 16. 0.44. 0.44. Okay, now this number, this one and this one, we express as allele frequency of capital A, and this one allele frequency of simple A. Now we have a method, we can calculate allele frequency of a population for certain phenotype. So then we collect all the genotypes, and from the genotype we can make a conclusion. This is allele frequency for capital A. This is allele frequency for simple A. For example, if you want to apply this for a human population, we take a easiest example. Take Mendelian characteristics in human. We have learned certain characteristics. Example, tongue rolling, then hitchhiker's finger like this thing. So then we can make a survey, and if we make the survey, so only thing is we have to analyze the genetic background. We have to analyze genetic level to check whether they have a homozygous or heterozygous. So this is a little bit different for us. If we know already recessive one, make the phenotype, then we can directly write the genotype, no issues. So simple A, definitely the genotype. But certain cases, if we see the phenotype, then we have a small issue. It could be capital A, or capital A, simple A. For that case, we can't say genotype. That means we need to analyze the genotype if we want to know it. Okay. Now, when it comes to let's say 100 years back, this is a hypothetical example. This is not a real one. And if we come 100 years back, we see more white flowers. Then what we can observe, 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 mean 10. 10 mean there are 20 alleles. Here there are 20 alleles. And 18, simple A. 18 is simple A. That means new allele frequency is 0.9. And this one is capital A is this 0.1. So allele frequencies change. This one increased. This one decreased. Capital A decrease, simple A increase. Okay, what is the message? Allele frequencies are changing with the time. They're not the same. So if allele frequencies change in the population, what we are going to accept, what we're going to take. So if you want to calculate allele frequency, should we do it like allele frequency in a given year? That could be one method. Then we have to make assumption. We have to calculate allele frequency only for particular years because it's changing. Okay, now we are coming to the Hardy-Weinburg concept. Now, Hardy-Weinburg principles, they introduce a method to calculate allele frequency in a population by making different conditions. That means we apply conditions when we are calculating allele frequency, because no one can make allele frequencies for every year. If a certain disorders, people want to calculate the allele frequencies, we have to use this number, not every year checking it. So we generally take a number, okay, this is the allele frequency of cystic fibrosis in UK. For that, we make assumptions as Hardy-Weinburg proposed. Okay, is this clear?
Yes. Clear, sir.
Yeah. So it is something like population is evolving. Hardy-Weinberg make assumptions. We'll see the assumption maybe after we learn this. Basically, the population is not evolving is one kind of assumption. If we think population is not evolving, then most of the concepts are fixed, because evolving population, allele frequency is changing. If you make a big assumption, no, this population is not evolving, then allele frequencies are not changing. So we can directly use these allele frequencies for understanding certain phenotypes. Okay, now Hardy-Weinberg equilibrium or Hardy-Weinberg principle, any kind of exam, they ask only two questions. One question is calculation. Second question is, what are the conditions used by the Hardy-Weinberg concept? Otherwise, we don't want to spend hours and hours to discuss about the Hardy-Weinberg concept. So simply, Hardy-Weinberg principle help us to calculate the allele frequency of a population. So somebody is challenging this one. How can you calculate? Because it is evolving, population is evolving, these are changing. Then we say, this assumption we made. Then the numbers are accepted. So we can clearly say population of cystic fibrosis in this country, allele frequency for the dominant one is this, recessive one is this. Okay, now there should be a question. Okay. Before we come to this question, there's a one point we can learn here. If we add allele frequency of simple A, so let's write like this. So if I add this allele frequency, I'm always getting number 1. So for example, this case, 0.56 plus 0.44, it's 1. In the second one, 0.1 plus 0.9 is again 1. That is understandable because if we toss a coin 100 times and we get a certain amount of that, out of 100. So if we add these two, total number is 100. So likewise, it is we are getting 1. And derivation of Hardy-Weinberg equation, which is also not required. So how they divide their formula or equation is also not required. We directly use the formula to calculate it. So if we use formula in physics or chemistry, certain uh, formula you are not going to bother about how the person derived this equation. So it's still, I remember like certain equations like PV equal nRT. In chemistry, these kind of equations, we just try to derive by Boyle's law and Charles, like these different laws we get, and we learn the concept and we can derive the equations. But here, it is not important. We can directly use the equations for calculations. Right, so we take a few questions. The frequency of two alleles in a gene pool is 0.19 capital A and 0.81 simple a. Assume that the population is in Hardy-Weinberg equilibrium. Calculate the percentage of heterozygous individual in the population. Calculate the percentage of homozygous recessive in the population. Okay, now the use of Hardy-Weinberg equilibrium to calculate the heterozygous individuals and homozygous individuals, number of people who are affected with disease, we can calculate many things. That's why it is still useful to evaluate in a population how many people are affected. So if we analyze these gene frequencies in a population, and if we keep the record, we have a approximate idea how many people are affected with certain genetic conditions. Then it's useful whether these kind of conditions are increasing or decreasing in a population. We can also check with the Hardy-Weinberg equilibrium. Okay, now to come to that point, what we have to remember, the Hardy-Weinberg equilibrium is, the equation is P squared plus 2pq squared q squared equal 1. So this is the Hardy-Weinberg formula or equilibrium or equation. Now what this P and what this Q? The P is dominant allele. Actually, it's a simple P, right? Simple P is a dominant allele. Q is a recessive allele. Simple P is a dominant allele. Q represents the recessive allele. Now, when we get a P squared, what is the meaning of P squared? P squared tells us this one. So we take a total in the population. When you take the total individual in the population, how many people having homozygous dominant? How many people having homozygous dominant in the population? That's a capital A example. So then P squared is exactly the number capital A divided by total. How many people in capital A homozygous dominant genotype in the total population? Now Q squared. Q squared is again total and recessive. So recessive in the sense simple A. So then we can write Q squared equal what is the total population divided by how many of them with homozygous recessive. Okay, then 2pq. So 2pq is equal how many total in the population and how many people are heterozygous. So this is a bit important now for us to remember what is the meaning of P squared, what is the meaning of 2pq, and what is the meaning of q squared. Now this kind of a simple calculation, how we can do it, capital A allele frequency is given, right? Capital A allele frequency is given 0.19 and simple A allele frequency is given 0.81. So if we add the capital A and simple A allele frequency, it should be 1. As a result, we can also write the second equation of Hardy-Weinberg, P plus Q equal 1. So P plus Q is the allele frequency of dominant and recessive. It gives always 1. Okay, now let's calculate the first one. Calculate the percentage of heterozygous individuals in the population. Now these numbers we can rewrite as a percentage. So percentage mean, for example, 19%, and this one is 81%. This is another way of expressing this number. First one, no space. I use the down space here. Calculate the percentage of heterozygous individual in the population. Now, heterozygous individual represented by, according to the Hardy-Weinburg equilibrium, 2pq. So 2pq is the heterozygous individual in the population. So we can directly use then the 2pq to calculate the heterozygous individual. So 2pq equal to 0.p is 19, q is 0.81. Point 19 multiplied by 0.81 multiplied by 2. 0.31 rounded up 0.31. Now this can also write 31%. So heterozygous percentage of heterozygous individuals is 31%. In this population, 31% represent heterozygous of this phenotype. Okay, next, calculate the percentage of homozygous recessive in the population. Now, this homozygous recessive equal to q squared. So, this equal to q squared. Now, what is q? Q is 0.81. So, this one's recessive one is actually q, and this one is p. So then second one, b, 0.81 squared. Uh, 0.66. So this is 66%. In this example, if we are provided the allele frequency of dominant one and the allele frequency of the recessive one, we can calculate the people in the population that they're having this dominant and recessive homozygous, recessive, heterozygous individuals like things we can calculate. This is a one example of Hardy-Weinberg calculation. So most of the calculations come into a fixed frame. That means there are no much different type of calculations. So they are always more fixed type of calculation. Right? So that's important to remember. What is p squared? What is 2pq? What is q squared?
So if we want to find the homozygous dominant, we can add A and B and subtract by 100, no? Sir?
You mean this percentage, if you want to calculate the percentage of dominant one, right?
Yeah.
Homozygous dominant.
Yes.
Yeah, 31 and 66. Uh, that could be a, 97, right? Yeah, 97. It could be then 3%. So, I mean, if we go approach that way, it should be 3%. Yeah. Or uh, if we can also go according to the Hardy-Weinburg equation, so dominant one is p squared, so it should be 0.19 squared. I mean, I did not use the decimals to a higher level, just rounded up. So this should be again 0.03, and it's a 3%. Okay, so another important thing, most of the calculations